Hi everybody, welcome to our first online lecture. We're going to be talking about eukaryotic replication initiation. A little high here, so I'm here. Okay. So just a quick basic mechanism underlying all replication is the addition of nucleotides, nucleotide monomer precursors that need to be added to the growing strand in a 5' to 3' fashion. Down here we have the template strand, as you can see. And so the nucleotide comes in, it's a triphosphate, so that means there's 3 phosphates here. After the reaction is catalyzed, 1 phosphate is ligated into the sugar phosphate backbone, as you can see, that's the new phosphate that we just put on. And then this is the byproduct, this pyrophosphate. And that happens in a stepwise fashion, driven by the complementary, the sequence on the template strand. In a kind of pretty simple way of summarizing it, there's 2 basic steps that need to happen. So again, we have our template strand down here, and it's sitting in the palm of the polymerase. And then the incoming DNTP has to come in, and that needs to get inserted into the new strand. Inserted, right there is our word for insertion. And then once it's in there, that's sitting in the active site. So that's that little piece, that new nucleotide needs to get popped over one spot so we can get the next template nucleotide in place for the next nucleotide to be added. So we have insertion followed by translocation of the template strand as each new nucleotide is added. So that'd be 1 insertion, and this was from last time too, so hopefully you remember, or it sounds familiar, plus 2 translocation. It's pretty ugly handwriting, huh? Okay, next. All polymerases have the same basic structure and some same elements in them. So on the left of our slide, we have a kind of more, I guess, a kind of a balloon model of a polymerase, and you can see it's fine to think of the DNA, any polymerase, DNA polymerase, RNA polymerase, Taq polymerase, RNA polymerase, like in SARS-CoV-19 virus, as a right hand. And on that right hand, there's a thumb, and then there are the fingers. And then the active sites down here in the palm pretty much are in the middle of the clenched fist once the fingers and thumb close down on the palm. Then down here, I think of this rightly or wrongly, is that little space on at the base of your palm going down to your wrist. And that's where the exonuclease domain is, which is critical for proofreading. So if the wrong nucleotide gets added, then it gets snipped out by the exonuclease, and the giving the opportunity for the polymerase to put in the correct nucleotide. On this side, we have a little bit more detailed image of a polymerase, and so again, we have our thumb here, the palm's down here, and the kind of at the base of it, and you can see the DNA entering through the middle, kind of running through the palm. And instead of just saying fingers, it actually specified multiple fingers on this model. So we have an index finger, a middle finger, and a ring finger, and a little finger, which are shown here in the different shades of brown and pink and off-white. So there's our 4 fingers and a thumb and a palm. So polymerase is our right hand. And I'd like to point out, we're eventually going to be talking about transcription factors. Transcription factors probably are the first place where the idea of fingers were described, as far as I'm aware of. And a finger is a type of domain that can literally interact with a protein domain that interacts with DNA. And so it's kind of small, it can kind of wrap around part of the groove. And so that are fingers. And we'll be seeing fingers again, zinc fingers, when we talk about transcription factors in a couple weeks. Okay, so what happens during replication in the middle of the active site? Basically, the hand kind of closes down on itself, almost like makes a fist. And so we have our templated G over here, and on this side where we're making a correct base pair, and then the incoming C is added. And since they match correctly, everybody kind of fits neatly into the clenched fist of the polymerase, and then we'll simply have translocation and make room for the next nucleotide to be added. And then you can see it might be an A, and if a T is matched against it, it fits nicely, inside the clenched fist, and this width, so let's do it in a different color just for fun. The width of CG and AT, I think that's a bad color because it doesn't show well. The width of CG and AT must be pretty close to the same because that's part of the fundamental symmetry that's seen in the DNA double helix. So that's just why it winds up so nicely and neatly. So what happens if we happen to be putting in a wrong base? It doesn't fit quite so neatly in there. So here a T is trying to bind with the G, and it's kind of distorted and it's poking out. Maybe that one of the fingers is not hurting. It's saying, ouch, looks not bind in there. It may get incorporated, and then this, since it's paired improperly, it'll get shifted down to the palm where that exonuclease is, and it'll get snipped off. And likewise, we can see the width is no longer the same, and it's pushing into the inappropriate regions of the tertiary structure of the polymerase. C and A, like if an A goes in, a C goes in and pairs against an A, again, you have things popping in the wrong spot, and things aren't too, are somewhat distorted in there and need to be removed. And like what? We have a great deal of distortion if we have two purines trying to bind against each other, because they're just much bigger and bulkier, so they're going to be rejected very quickly. So these are the mismatched ones, and from these all go down to the exonuclease domain and get removed. Exo. Remove. Okay, that's that. Now we can take a look at some replication forks. So over here on the left side of the slide, we have the fully assembled E. coli replication fork, and we can see there's the Pol3 coenzyme here, here, and here. We have primase, which needs to make priming, an RNA primer on there. We have our good old friend, the clamp loader. We have the sliding clamp. We talked about all these. I'm not going to go into this in any great detail right now because there's that really great video, and I added it to our course resources, which I think explains it very clearly, the assembly of and the processing of DNA replication in E. coli. On this side of our slide, we have our new friend, the eukaryotic replication complex, and you can see structurally it's quite similar. We got our polymerases. They may or may not be a little bigger. We have RFC and PCNA, which are comparable to the clamp loader and the sliding clamp. We have a few more proteins involved here that would probably help some processivity and regulation. And what we're going to do now, oops, let me just mark there. So here's our this clamp loader, this clamp loader, another clamp here and here. We got our polymerase epsilon and delta, and we'll talk about them in a few seconds. And we got this important guy MCM7. We're going to be talking about him for quite a bit. And instead of having primase, we have polymerase alpha acts as primase. So all the same functional elements are there. Structurally, it's pretty much the same. It's much more refined, and we have a bigger genome, so we can have more players involved to give her higher efficiencies of regulation and specificity of regulation and things like that. Okay, so this table here just summarizes and sets you up for your homework. The proteins involved in replication in E. coli, right there. And again, I'm not going to go through them in detail, but you can see some that hopefully look a little bit familiar from 2.5 weeks ago. And then this next slide here is a comparable list, but it's the proteins involved in eukaryotic replication, as you can see there. And again, there's a long list of them, and obviously there's going to be some similar functions. We saw similar structure. We know the purpose is the same thing. The functionality is the same thing. And with like the homework, you'll see I'd like you to kind of map out which proteins are doing the same thing in the different systems. Okay, so let's talk now about eukaryotic replication in yeast. And particularly what we're going to be talking about is not the addition of the nucleotides, but the regulation and the activation of it. So this figure is taken from your textbook. There's two basic steps involved in that we're going to talk about here. One is called licensing, and one is called activation. Licensing is kind of tagging replication origins so that they're ready for replication to occur when that time comes in the cell cycle. This is the licensing part shown up here in this half of the diagram. And then activation is the transition from that licensed saying, I can replicate, I'm good at replicating, to turning that replication machinery on at the right spot at the right time. So let's see what's next. Okay, so now let's take a look at an ARS element in yeast. One ARS stands for autonomous replicating sequence. So let's make a mark here. Autonomous replicating sequence. Each ARS is between 100 to 200 nucleotides long. 100 to 200 nucleotides long. And it's composed of 4 sequence regions, domains. Okay, so we have an A domain, we have B1, B2, and B3. Within the yeast genome, there is about 400 ARSes. They're located every 30 kilobases apart. And as I said, they're 100 to 200 base pairs long, with these elements required to make one functional ARS. So when we call something an ARS, that means that if we were to take a plasmid and put an ARS in it, that plasmid would be able to replicate in yeast, because ARSes are functional in yeast. Similar to the way when we make plasmids for E. coli, if we put a ORI sequence in a plasmid, then that allows it to replicate in E. coli. So plasmids that replicate in yeast must have ARS sequences in them. So what are the functional elements within the ARS? Well, we're going to be talking a little bit about this guy in a few minutes. It's called the origin replication sequence, origin replication complex. And that is a protein complex that we'll look at on the next slide, and it binds in a region that's composed of A and B1. So it's going to bind right up here. These regions, these elements B2 and B3 are described as helically unstable, suggesting that they're AT-rich. So that's generally what makes things helically unstable. Rich. And then within the A element itself, there's a specific 11 base pair sequence that's called the ACS, which is 11 base pairs long, that is AT-rich, extremely AT-rich. Because it's only 11 base pairs long, there's actually, within the entire yeast genome, there's 12,000 copies of it. Oh yeah, ACS stands for ARS consensus sequence. Okay, so even though there's 12,000 copies of it, within the genome only 400 of them are functional. Those are the 400 that are actually within the ARS, the completed ARS element itself, so there's a few extras. So when we talk about ARSes in other eukaryotes, there's a couple things that yeast is nice because it's small and it's relatively simple. Oops, sorry. But ARS elements in other eukaryotes are larger. They're not as well defined. So an ARS element in yeast is this 1 to 200 nucleotide base pair long region. In other eukaryotes, there are lots of ARSes, and they're much more, their sequences are a little bit more complex. They're not necessarily only 1 to 200 nucleotides long. So their sequences aren't as well defined. They tend to be larger. There's a lot more regulation that needs to go on in other eukaryotes because there's different types of coordination between transcription and different cell types that needs to happen. In yeast, where we have this 1 to 200 base pair long arse, that's about the size of one nucleosome. So once the arse, the pre-replication complex gets assembled on here, it's pretty much not a nucleosome per se any longer. In other eukaryotes, they tend to have fewer nucleosomes and are likewise tagged in some manner, but over a larger stretch of DNA. Okay, so now let's talk about how a pre-replication complex assembles on top of an arse. So the first player that is involved in assembling onto the pre-replication complex is this protein here, ORC, which is origin replication complex right here. It's a hexamer. The proteins within the hexamer have the unexciting names of ORC 1, 2, 3, 4, 5, and 6. The binding of ORC onto the arse is an ATP-dependent process. And ORC binds, as we said on the previous slide, in a region defined by A and B1. Once ORC binds, we recruit two more proteins, which are shown here, CDT1 and CDC6. CDT1 and CDC6 together in turn recruit the binding of MCM2-7. So once the full pre-replication complex is assembled, you can see that there's one ORC kind of anchoring the whole thing, and then we have two copies of CDC6, two copies of CDT1, and then two copies of MCM27. This licensing event happens in early G1 or in late mitosis, and therefore the licensed ORC is marked for most of the cell cycle. It's sitting there, it's inactive, its replication is not occurring, but it's tagged as a place where once the proper time comes, the cell knows where to initiate active replication forks, which are marked here by the MCM27. The MCM27 themselves are each also hexamers, as you can see over here, hexamers. And basically, the replication assembles until it's bidirectional. The replication forks are in a replication bubble that's bidirectional. And each of these marks the location where helicase is going to assemble and start driving the initiation of replication bidirectionally, out, extending away from the ORC. Excuse me, extending away from the arse. Okay. So your bidirectional replication bubble is initiated at the arse, defined by the ORC, and then the dwarves. No, just kidding. Okay, here we go. That's how that works. Okay. So here we have our pre-replication complex that got assembled there. So now we want to activate this guy. So what's going to happen? We have two kinases are going to come in. One's called CDK and one's called DDK. And I'm going to talk about them a little more specifically in a few minutes. But what they do, what's going on? What they do is they actually dislodge CDC6 and CDT1 and ORC. those guys are now dislodged off of here, and they make room for all these other proteins, many other proteins, to assemble on top of MCM7. MCM7 is not going anywhere, and this doesn't even list all the proteins that assemble on top of MCM7, but these proteins, CDC5, GINs, plus other proteins not shown, form what's called a pre-initiation complex. And that, again, also requires lots of ATP, and this pre-initiation complex ultimately is converted to what's called the CMG helicase, or an activated helicase. So some of those pre-initiation complex proteins disappear, but MCM stays around, CDC45 stays around, and GIN stays around. And those three together are the CMG helicase, or the CMG complex. And they function as the helicase, which unwinds the DNA at the heart of the replication fork. Once DNA is unwound, then it needs to be replicated, and that's where the polymerases come in. So we have 3 polymerases that are primarily involved in replication. In the absence of mutation, we have polymerase epsilon, which drives replication of the leading strand. We have polymerase delta, which drives replication of the lagging strand. We have polymerase alpha, which is the primer. It primes, makes little RNA primers so that the other polymerases can do their work. So let me erase that and that and that. Okay, so we went from G1 pre-replication complex to pre-initiation complex to CMG helicase to... So we've now gone from the pre-initiation complex, we've had the helicase assemble here, and we have our fully completed replication fork shown here. This picture is not from your text. I'll show you that one in just a second. I think this one's labeled a little bit more clearly. You can see the CMG complex I talked about right here. And with the component proteins of CDC45, MCM27, and GINs. This GINs is an acronym from Japanese. I forget exactly what it stands for. I can look it up for you later if you're really interested. It's some Japanese thing. It's 5143 in Japanese numbers. So what else do we have here? So we have our epsilon. Our polymerases are shown here in the replication fork. We've got epsilon, which is replicating the leading strand. We have polymerase delta, which is replicating the lagging strand. And since we're making Okazaki fragments on that lagging strand, we're continually priming a little bit right here with polymerase alpha. We have to protect single-stranded DNA wherever it's exposed, so we're just showing some representative single-stranded DNA as it's being protected by RPA. We have the clamps associated with each polymerase, which help with processivity, and a couple other proteins I'm not going to talk about. So let's take a look at that now in our version from our textbook. And so just to take a quick look at what the version looks like in your textbook, that's why I kind of prefer the other one. It's labeled a little more clearly, but again, you can see, let's see, we got our MCM27, we got some pCNA, we've got our polymerase right here. I don't know if it shows it unless I drag it. Here we got clamp loader, right? This guy's the clamp loader. This is the clamp. So we've seen lots of these elements in the eukaryotic replication fork. Okay. So I want to remind you, we talked about this at the end of talking about nucleosomes and chromatin, that we talked about the epigenetic modifications that happen to nucleosomes, and which means that they get marked in some manner so that during development they get marked as either a gene that's been turned on or turned off. And so during replication we want to maintain that knowledge in that region of the genome. So that's what is referred to by marked, and we talked about acetylation and we talked about methylation, and there are some of the ways that histones can be marked. And it's important that as replication passes through an area that the histones reassemble with the correct mark on them so that that information isn't lost in that region of the genome. So this is the same slide we had earlier when we talked about chromatin. I just want to remind you. So as the replication fork passes through and the nucleosomes had to be disassembled, The marked H3 and H4 are assembled right away as soon as replication is done. It's very important to get those nucleosomes repackaged just for their protection and for their packaging, and also to maintain whatever regulatory marking has been put on them. And so we saw that the chaperone protein CAF1 helps histone H3 and H4 assemble, and then the chaperone protein NAP1 follows up with H2A and H2B. And H4 may only transiently disassociate from the strand. H2A and H2B are thought to be hanging out in fairly high local concentration. So all those factors contribute to maintaining the mark of the epigenetically marked chromatin as replication passes through. Okay, so the last thing I want to talk about is how eukaryotic replication is coordinated with the cell cycle. Obviously, we know by definition that replication occurs during the S phase of the cell cycle, but there's a lot of regulatory work that goes on to maintain that proper timing. And there's a class of proteins called cyclin-dependent kinases, which are involved in regulating replication with the cell cycle and regulating the cell cycle in general. So what cyclin-dependent kinases are there working in the process of DNA replication? Well, let's see. Here I've marked that we have 2 of them are shown right here. This is actually in the activation phase, right? Because we have our pre-replication complex up here, and we talked about how CDK and DDK are involved in dissociating these 2 proteins and allowing the assembly of a pre-initiation complex. So CDK and DDK are involved in activation. What are CDK and DDK? CDK is a cyclin-dependent kinase. CDK2. And DDK is a dimer, has one subunit here called DBF4 and a second subunit called CDC7, which is another cyclin-dependent kinase. And if we even backtrack from activation just to licensing, we can see up here, in this licensed arse up here, we have a CDC6, so that's yet another cyclin-dependent kinase that's associated with licensing. And if we go down here to the active helicase in the active replication, we have CDC45. So we have already several cell cycle related proteins that are marking various steps of regulation and activation and ongoing active synthesis of DNA. Hopefully that's not particularly surprising at all. Let's now take just a little review look at where those steps are in a cell cycle. Here now we've transitioned to blowing up the cell cycle image itself, and I just want to highlight a couple of the cyclin-dependent kinases we just talked about. So here's your CDK6 or CDC6, which is, as we know, on the licensed origin. We have CDK2, which is involved in activating the origin and taking it from its licensed state to its activated state. And then during replication, we talked about the fact again, it's not marked up down below, but it's marked up here, that we have CDC45 that's part of the helicase, the CMG complex includes CDC45. So this important coordination of replication with the cell cycle has to be maintained. Whenever that's lost, that's part of the process of a cell becoming cancerous, when we lose that coordination between DNA replication and overall cell growth. And I think that's where we'll stop with this material about regulation and initiation of replication. Talk about replication with that. So let's say that for your homework for this set of slides is to compare and contrast the E. coli and the eukaryotic replication initiation process and the machinery. That'll help you review the old E. coli stuff. And kind of compare it and see the similarities and the differences between Eukaryotic and prokaryotic DNA replication. Machine. Hurry. And I'll put a Moodle assignment for that. And you can either make a table or you can write it out by sentences. There's a lot of proteins. I think you got a lot to work with those tables in this talk that are in your textbook, and the structures that we talked about. And so that's the end of replication initiation. Our next talk will be about replication termination, and specifically about telomeres and the so-called end problem that happens in linear chromosomes. And with that, I will sign off. See you soon.